In addition to maintaining optimum pH, in addition to maintain optimum temperature, we have to maintain optimum pH as well as osmolality. We discussed earlier about temperature as well as osmolality. 300 milliosmol is sufficient for the growth and development of the cell in the cell culture. Same manner in the case of the optimum pH maintenance is done with the help of buffers. Why we have to add buffers for the growth and culture media is the reason is that when the cell grows and divides, that time some metabolic acids as well as alkali will be produced. In order to buffer these excess of acids and alkali, we require a natural buffering system. Normally in our body we have variety of buffers are available, that is carbonate buffers, phosphate buffers, and ammonia buffers are available. And here in the case of the cell culture, we require the natural buffering system such as bicarbonate buffer system that is utilized for our purpose. And the pH is interrelated with the increase in the level of the carbon dioxide, because when the cell grows and divides, oxygen is supplemented to the cells, and during the metabolism carbon dioxide is generated. That carbon dioxide is converted into the form of carbonic acid in the presence of carbonic anhydrase. Further, this carbonic acid degrade, that is breaks down into proton as well as bicarbonate. This proton and bicarbonate, the proton contributes for the pH and bicarbonate is buffering the acids which are present in the medium. So this is an excellent buffer which is present in our system, that is also utilized in the growth and culture of the animal cell as well as human cell. Nearly 5 to 10% of carbon dioxide is usually maintained in the carbon dioxide incubator. Externally, within our body, the carbon dioxide is generated and it is buffered by our own system, but in order to maintain the normal condition of these 5 to 10% carbon dioxide, we require a special system, that is an instrument called as carbon dioxide incubator. And moreover, this natural buffer is lower in cost and non-toxic. It doesn't produces any byproducts. The presence of pyruvate in the medium results in the increased endogenous production of carbon dioxide by the cells. Thus, the pyruvate is advantageous because the less dependency of external supply of carbon dioxide. So, normally the bicarbonate buffers follow the Le-Chatelier's Braun principle, that increase in acidity in the medium is established by increase in the proton formation, and the free bicarbonate ion reacts with the excess of this proton to form the carbonic acid. That carbonic acid shifting the reaction towards the back of carbon dioxide and water formation, and that is it decreases the proton concentration, and naturally it shifts that to the right of this. Chemical buffering. In addition, some system we add chemical buffers too, that is EP's buffer. That is, the chemical buffer is a zwitterion, that is 4,2 hydroxyethyl 1 piperazine ethane sulfonic acid is utilized as the superior buffering, which is having a capacity of pH range around 7.2 to 7.4. This is a EPS is having a controlled gaseous atmosphere, is not required for it. It relatively expensive and toxic at higher concentration for some cell types. So it cannot be utilized for all the cells. Only few cells can be utilized for this because of its toxicity on higher concentration. Another buffer, chemical buffer used is nothing but phenol red. The phenol red is commercially available in media contains this pH indicator. This is effectively used to monitor the pH range during the metabolic activity as well as during the cell growth. When the phenol red is present, the media changes its color as the pH changes. Normally below the pH 6.8 it is yellow in color. When the pH increases to 8.2, it changes from yellow to pink in this case. So by observing the color change, we can able to identify the changes in the pH by the help of phenol red. Normally, the optimum pH for the cell growth is 7.4. At that time, the media containing phenol red will be bright red in color. So, from the bright red color, we can see it is an optimal pH is maintained. So, when any change when it deviates from the bright red to a yellow or to pink, it's an indication that there is a changes in the pH. So there are various equipments which are used for our cell culture. We come across to maintain the proper temperature, carbon dioxide for the mammalian cells, and humidity of about 95%. A different shape of the cell culture flasks are available that we include. What are all the different culture vessels which are utilized for our purpose is nothing but Petri plates, culture flask, multi-well plates of several sizes which varies in the diameter and as well as in the shape are utilized for the purpose of adherent culture. What is adherent culture means? And further we make use of the cell scrapers to remove the cells which are adhered to the substratum, that is the culture vessel. So normally an ideal cell culture laboratory should contain the equipment such as laminar flow hood, carbon dioxide incubator, inverted microscope, autoclave, centrifuge, etc. So these are all the various equipments. Let us discuss in detail about the laminar flow hood or biosafety cabinet. Normally the animal cell cultures are handled at entire process in a aseptic manner, that is without any bacterial or fungal contamination. So always aseptic condition is maintained with the help of disinfectants that provides a controlled environment in which the microbes and contaminants are eliminated from the media as well as in the culture flask also. The laminar flow hoods are acting under air under negative pressure. That is, we can see it is fitted with the HEPA filter. Already I told you 99.9% of the air, that is any entrance into the laminar flow, is filtered by this high efficiency particulate air filter that cleans the air which is traveling at a velocity that can be calculated and prevented from the intrusion of the unfiltered air which is prevailing in the work area. The primary filter is present at the top of the laminar flow hood. Then further, the HEPA filter is responsible for filtration. Some cabinets are operating at a filter that is sterilizing the air taken in, then excluding the larger particles from the bacterial as well as fungal organisms. So that may create contamination of the culture what we are using. There are two types of laminar flow hoods are available. One is horizontal as well as vertical flow. Horizontal means the flow of air that occurs through the horizontal position and it goes in the upright and it is going the exhaust, just like a chimney which is present in the house. The flow is there from the bottom, from when we cook from the cooking area, the vapors goes in the top, it rises above and it goes out through the chimney. Same manner the horizontal flow will be there. Whereas in vertical laminar flow chamber, what happens? The air enters from the top and it flows through the working area of the laminar flow chamber in this case. So it is filtered by the HEPA during the inlet in the whatever may be the position, whether it's an horizontal or it may be a vertical. In the horizontal cabinets, laminar fluid, air fluid is fitted with a fiberglass screen that prevents the entry of particles, as well as exhaust fan is also fitted with that, so that it pushes the air which is present inside so that purified air is circulated by the EPUF filter, which is fitted at the bottom of the laminar flow hood. Whereas vertical flow hood, in which EPUF filter is fitted on the top and filters the air that is present at the back of the laminar flow hood, and so that the air is coming and going out in a proper manner. The vertical flow hoods are most popular when compared to the horizontal flow hood, because it doesn't have any proper protection in the case of the horizontal flow hood for some sort of culture, hence it is not widely used when compared to the vertical flow laminar hood. And next instrument what we are using for our culture is nothing but carbon dioxide incubator. The carbon dioxide incubators are in need of external supply of carbon dioxide to maintain the fixed level of the carbon dioxide in the incubator. In order to maintain the carbon dioxide level in this carbon dioxide incubator, that is 5 to 10% of atmospheric carbon dioxide has to be maintained, that is in turn provided by the carbon dioxide cylinder that are attached with the incubator for this purpose. And normally it is having a highest relative humidity of minus 95% for the growth of the medium. Normally the carbon dioxide which is supplemented by the carbon dioxide incubator, that is converted to the bicarbonate buffer with the help of our carbonic anhydride, which is present in the cells. It's a natural buffering system that is maintained by the culture. Another equipment which is used for our cell culture is nothing but our inverted microscope. What is inverted microscope means? This inverted microscope has phase contrast optics, and it is responsible for the identification of the cell, degree of spreading, membrane, blebbing. All these things can be analyzed with the help of this inverted microscope. The cell morphology is very sensitive, and any alteration in the cell media, as a composition of media, or osmolality changes, or any changes in pH and temperature, that affects the morphology of the cell, that can be easily spotted. And further, any contamination when it is present in the cell culture, that can also be identified with the help of our phase contrast microscopy. Here we can see the light source which is on the top, and the field aperture is over there, and we have the ocular or eyepiece for the viewing purpose, and diopter adjustment to get the clarity in the cell culture, and is the stage, and is the beam splitter is available, and the objective lens is found at the bottom of the stage, and coarse and fine adjustment for the clarity of the picture, that is for the purpose. And lamp is also fitted with this. The lamp provides illumination from the bottom of the culture vessel. It may be the culture flask, flat culture flask, or it may be petri plate. That's all about the various equipments we discussed. What all the equipment means: laminar flow hood, carbon dioxide incubator, inverted microscope, and various culture vessels are utilized for the process of culture. We will discuss about the types of cell culture in following video.